Okay, I think we can get started now. Good morning, everyone, and thanks for joining the webinar. My name is Will Cronister. I'm a senior computational scientist here at TokuProfile. And if you're not familiar with us, we are a company focused on immune profiling using mass cytometry and spectral flow cytometry. And with each project that we do in collaboration with our clients, we offer three analysis tiers, which are base, core, and premier. And today I'm going to walk through the differences between these packages and the features that are included within each one. And to do that, I'm going to show you those features on our interactive web app called TokuProfile, which you'll receive at the conclusion of your project, and as well as for updates throughout the course of your project if you choose to work with us. So let's get started. I'm going to share my screen, and while I bring up the web app, I just want to encourage all of you to submit your questions as they arise throughout the presentation, and I'll be happy to answer those at the end of the webinar. So I'm here on the TokuProfile web app. This is our demo page. This is freely accessible to anyone. You can go to our website, and there's a link to the TokuProfile, and you can log in with demo credentials and look around at this for yourself. But today I'm going to look at a project we did with the University of Minnesota. This was a study having to do with acute graft versus host disease. As you can see on this landing page, we have an overview of the goal of the study and some key questions that the researchers were interested in answering. The base tier that we offer with our base level analysis includes this page, as well as Samples tab and Data Export tab, if we go on this left sidebar here. The core features will expand your access to the gated analysis section, including a lot of visualization tools, as well as statistical testing that allows you to compare between different groups within your study and get an idea of any significant populations that are changing over time or changing between responders, non-responders, that sort of thing. Finally, the premier tier expands your access to the unsupervised analysis, which is our unsupervised clustering. And this is an additional branch of analysis that is complementary in some ways to our gated analysis. Whereas our gated analysis is more of a traditional gating approach, where we have a gating scheme that's pretty well established, and we follow the typical rules for gating cells into smaller subsets. Our unsupervised analysis takes a bottom-up approach, where we cluster the data and see what cells our algorithm associates with each other, and then we annotate those and try to figure out what those subsets are. So start with the base features. As I said, we have this landing page, this overview page that summarizes what the study is. This is all done in collaboration with our clients, obviously. And below that, we have the subset hierarchy. And so this is essentially the gating scheme that we use to take the mass cytometry or spectral flow cytometry data. We start with a top gate of live cells, and then we break that down into smaller and smaller subsets to get more characterized subsets, looking at the subsets like B cells, B memory cells, B naive cells, and so on and so forth. You can see there's some nice granularity here to characterize the populations within your data. And to the right, these check marks indicate state markers that are expressed within these subsets. And so we also check for the percentage of cells within each of these subsets to the left that express a particular marker. So, for example, AREG was one marker that these collaborators were interested in. And so this was assessed in B cells, B memory cells, and so on and so forth. So as you can see, there's quite a bit of data that you get from doing this analysis. You get the percentage of expressing cells within all of these subsets for all of these markers, anywhere that there's a check mark. So this is all displayed for you as a quick guide to the data that's contained in what we deliver to you. There's also a reports tab here, which for this project, there's nothing here, but for your project, there would be a sample and gating QC report here that gives you an idea of the quality of the samples that we received and any information and details about the gating that we did on the data, which you're free to review as soon as it's available. You just log in. We post the report for you here, and you can review it on your own time, download it, send it wherever you want. Moving on to the samples tab, this is a place where we summarize the samples contained in the study. So in this case, we had 40 samples from 20 subjects, so 2 samples per subject. You can also see more detail about the data in this list of samples. We have sample names from University of Minnesota, so they can go off their identifier, or we also have our own internal identifiers. So we have an easy way to identify each sample. And then if you scroll to the right, there's more metadata that's provided to us by the client. All of these individuals were acute graft versus host disease. And then their responder status is here as well. So responders and non-responders. We have details about the treatment they received, as well as some additional data on the plasma levels of ST2 and AREG that were of special interest in this study. So whatever metadata you want to provide and use as a lens to look at your data, or a way to divide your data into groups for statistical purposes, that can be provided, and we can run statistics breaking down the data whichever way you want. And that's illustrated here in these boxes. For example, looking at the study time point and the response. So we can look within baseline samples. Are there differences between non-responders and responders? That might be really interesting to see. And then on treatment, we can look at differences between non-responders and responders. And this can give you some really helpful insights into why a treatment is working or why it's not working. Moving on to the data export tab. This is where you'll get your raw data, which is an FCS file in the case of cytoph and spectral flow cytometry. So these files are available for download. You can download them all at once, or if you only want one or two, or any number, you can check them and export. And this will go to our downloads section, and it should populate pretty quickly. And then you can go ahead and download that to your computer. So we try to make it as easy as possible for you to get your raw data, as well as the gating ML files, which are basically the guide to the gating that we do on the data. So how did we get from all these live cells, this big pool of cells, down to CD4 T effector memory cells? You can reconstruct and sort of retrace our steps of what we did to get there using these gating ML files as a way to reproduce our methods. The data tables are another really useful data type. So this is where you get essentially the processed results of our gating. So if you have gated data, you can get population frequencies, which are your T cells, your B cells, and so on. Your cell state subsets, which are the percentage of cells expressing a certain marker, like AREG, like I pointed out before. And then finally, the marker expression data, which has to do with the intensity of expression of these cell state markers. So not simply the percentage of cells expressing something like AREG, but the intensity of that expression, which can vary in interesting ways between responders, non-responders, different time points, things like that. You can choose any of these and download them. You can append the metadata if you'd like to have that available within the same file. And this will export as a CSV. And just to quickly show an example, I can pull this up in Excel. And what you'll see is we have these three columns at the beginning. This is without the full metadata appended, but it does come with information about the sample. So each row here is a different sample and a different population. And what we're looking at here in the second row, the first one of the results, is we're looking within a top-level population of non-granulocytes. This is sort of the parent population for all of the interesting cell types we focus on within a PBMC sample, for example. So B cells, this is looking at the population of the number of B cells within this parent population of non-granulocytes. And what you can see is we have about 2,000 B cells for this sample, and that was out of a total event count of about 200,000. And so that works out to roughly 1% of non-granulocytes were B cells. And these are the numbers that this comes out of our gating process, and you can use all of this data here. We have every population in here: B memory cells, B naive, plasmablasts, and so on down the list. And you can take this data and easily port it into graphs if you'd like to plot your data, prepared for figures for presentation to colleagues and so forth. We try to make it as easy as possible for you to take the data, the results of our analysis, and take it and run with it and do whatever you want with it. So switching back to the web app, we also have, like I said, the cell state and the marker expression data available here, and you can export all of that data very conveniently. If you have a premier level project, I'll just mention the clustering data, which we'll get to later, is also available on this data export page. But if you're only doing a base level project or a gated or a core level project, you'll only receive the gated data. And then the metadata is also here for you to download for your reference if you'd like. So this concludes the base section of the features. This is what you get. This is basically, you get the results of our gating, and we can answer any questions you might have about that in our collaboration, but this is basically what you receive with a base tier project. If you have a core level project, you, like I said, get access to this gated analysis tab. And so gated analysis is going to open up all sorts of ways to visualize your data. And we're constantly working to improve these methods as well. So these will only get better and better with time. But what you can see from this plot is this is a display of each major cell population within the gating scheme. And each dot here is a sample. So we have roughly 40 dots per subset. It should be 40 if every sample has some level of these cells. And what you can see is that these are arranged from greatest to least, so monocytes and classical monocytes, and there's some overlap between those two. Those are the most prominent populations, and then it goes on down the list, natural killers, CD4 T cells, and so forth. And you can do a lot of different things with this. This will be a recurring theme throughout these visualizations, but we try to make it easy for you to group things by response and get a quick look at any interesting trends, any interesting differences between, for example, non-responders and responders. You can also filter based on any metadata you'd like. If you only want to look at baseline time points, that'll drop out about half the data points here. And you can hover over any of these data points to figure out which cell is, really high in CD4 T cells. Again, we try to make this as easy and convenient for you to glean insights from your data and take that and maybe come up with new hypotheses or new ideas for further downstream analysis. We also present this data as a stacked bar plot. So now we're viewing the data where each vertical bar is a sample, and then each colored chunk of that is a population listed to the right here. And again, we aim for this to be convenient and interactive. So if you want to, for example, uncheck the CD4 T populations and look at everything else, you can quickly do that and get a sense of what proportion every other cell type takes once you filter out the CD4 T cells. Moving on to group similarity, this is a PCA plot. This allows you to get a quick look at how the data are separating based on the immune populations that we've gated. So again, another interesting thing to look at might be coloring by response. And what you can see is there is actually an interesting separation here between responders and non-responder samples along PC2. You can also view the contributors that are driving these separations and see which populations are contributing most to these different PCs. Moving on to the immune changes over time tab, this is another really helpful tool for visualizing data. So we have 3 subtabs here, which are population frequencies. This is, again, your B cells, your B subsets, your CD4 T cells, and so on down the list. And so you can select all of these, and it'll make a plot for each one, looking at the base time and on-treatment time points, as this is the immune changes over time. So we have time point along the x-axis here by default. You can also look at the cell state markers. So this is within each cell type and within each marker. You can observe any differences here. And so what this is indicating is, for example, the expression of CCR7 within B cells. What percentage of total B cells express this marker? It's pretty high in both categories, but there are other examples where one might be one group one time point baseline or on treatment might be higher than the other, and this FDR value is the multiple tested corrected p-value for our statistical testing, so you can quickly get an idea of whether there's a significant result. Marker expression is another data type. As I mentioned, this refers to the amount of the marker being expressed. And so here I'm going to highlight Tregs and look at all markers within Tregs. And what you can see as we scroll down There are roughly 8, there's exactly 8 plots per page. And so there's multiple pages of results here. The Tregs, you can see there's an interesting result here where Tregs at the baseline time point have a higher expression. This is the median expression of the marker CD38 at baseline, and then on treatment it declines significantly. So this is indicated by the FDR value. And there's also a way to filter this. So if you want just the significant results, some of these pages have hundreds of plots for you to review, but you can always filter by a cell type or a marker, or by the false discovery rate to get to just the most interesting results and save a lot of time that way. CD38 and Ki67 are both showing significant decreases between baseline and on treatment here. Moving on to immune differences, this is where you might look at endpoints other than time. So one endpoint of interest might be response. So you can indicate that and then select all cell types. And this will now group the data by non-responders and responders. And so here you can look for significant differences between non-responders and responders. If you want to look within a certain time point, for example, on treatment, you can indicate that in the filter step. And now we have some statistics showing up that we can filter on. And you can see there are several populations that are significant or trending close to significance for differences between non-responders and responders during the on-treatment time point. So this is a quick way to get to, again, interesting results that might drive further downstream analysis and follow-up studies to see why it is that these CD4 T cell and CD8 T cell populations are associated with response. You can also normalize these values to baseline. That might change your results somewhat, but the option is there. I'll quickly unselect the false discovery rate and just show what this looks like. So now we're looking at it normalized where the baseline time point is all set to 0, but these values are set in proportion to that. So that can be another convenient way to sort of set all of your subjects to the same baseline point as 0, and then look at the changes from baseline at a later time point. So this is where the core features end. This is what you get with a core level package. And I'm going to move on to the premier level results now. So this is our unsupervised analysis. And in many ways, it's parallel to the gated analysis, a lot of the same visualizations, but what you're seeing here is not cell populations per se, but clusters. So this is our unsupervised clustering results. These are the names of these clusters are referred to the annotated cell types that we assign to the cluster, as well as the cluster number to differentiate any clusters that might have the same annotation. And what you'll see is that there's actually more clusters than cell populations that we saw in the gated analysis. And that's because our clustering algorithm, I believe our gated analysis had about 31 populations. Our clustering analysis found 41 populations here. So there's 10 additional clusters that indicates that many cell populations that we gated as being one group might have subpopulations within them that express markers in different ways. And so to illustrate that, we can look at the cluster heat map, and we can look at, for example, the CDT clusters. So this is any cluster that was labeled with a cell type that falls within the CD4 T cell category. And what you'll notice is that there are 3 CD4 T effector memory cell clusters labeled here, number 9, 21, and 18. And what's interesting about this, when this sort of thing happens, is that you can sort of look at the heat map and figure out, okay, well, why do these cell types, even though they all resemble T effector memories, what makes them different such that our algorithm splits them into 3 clusters instead of 1? But it seems to be driven a lot by certain markers being expressed. So HLA-DR, PD1, CD25. So in general, when this happens, we're looking at perhaps a subset that's activated compared to the other cells within the same category, or cells that might be exhausted. So there's lots of different interpretations you can make about why these clusters separated. And these can be some of the most interesting ones to follow up on with statistical analysis, because if these unique clusters are associated with a certain functional status of these cells, that might indicate something that's associated with response or time point, or if your study features different dosage levels, that might be something to follow up on as an interesting biological mechanism. We also have a UMAP. This is a very common technique within single cell analysis. So every dot here is a single cell, and we can color this by response, just to get a quick at-a-glance view of where the responders and non-responders differ from each other. Some cells that might be interesting to follow up on would be areas where there's a lot of blue and not much red, or there's a lot of red and not much blue. So there's quite a bit of overlap here, but there are some interesting differences that might be worth following up on. And that will, of course, be reflected in statistical testing that we do as part of this analysis as well. Moving on to group similarity, we have a PCA plot again. And likewise, we have immune changes over time, although here we are again looking at population frequencies within clusters rather than our gated populations. We can normalize these to baseline, and we can also connect time points by subject. This is another feature that I forgot to mention earlier, but this is available to give you a direct look at how things are changing between baseline and on-treatment time points. And then finally, immune differences, you again have the ability to separate the clusters by responder status and view those results and potentially find some interesting significant differences. With that, that concludes the premiere, and that's a wrap. As you're thinking about doing a project with us, you might want to consider whether you have resources in-house that can do a lot of this analysis. If you feel confident that you can get everything you need out of just the data, maybe a base level project is right for you. But if you want to leverage our skills and expertise in the software we've developed to analyze this kind of data, we recommend you consider a core or premier level project to get the most out of your data, and we'll do our best to pull every interesting insight out that we can using our well-validated pipeline. With that, I will sharing, and if there are any questions, we can get to those. If you have any questions right now, please feel free to send them in. I have a couple in front of me. Okay, can I upgrade my analysis tier? Yes, so in a situation where you purchased a base level project, you can always upgrade to core or premier if you so choose. We always have that flexibility. There's nothing set in stone up front. Once we collect your data, we can then extend the analysis to include that statistical testing and expanding your access on the web app to these visualization tools. As well as the premier level tier, we can run unsupervised clustering for you and get those results integrated into the web app as well. Yes, absolutely, you have the option to upgrade your analysis tier if you so choose at a later date. If I purchase the base tier, is it possible for me to do the analysis provided in core myself? Yes, like I was mentioning, let me just share again. The base level tier includes essentially the raw data plus, so everything in the data export tab is really the most useful for, these are the key deliverables for a base level project. So we have our FCS files and gating ML files, so you can sort of reconstruct and retrace our steps with our analysis. The data tables, you will have the gated data, all three of these subset levels, as well as the metadata. So yes, we will make everything available to you. All of the values that go into our gated analysis are available within these downloadable data tables. So if you, like I was saying, if you are from a very well-staffed department where you have a data team that's ready to handle this data and do everything you want with it, that's perfectly fine. For customers that don't have that available or want to save their own team some time, that's why we offer the core and premier analyses to pull out those useful insights that can drive your study forward. Again, it comes down to your preferences, your budget, and what insights you want us to pull out, or which ones you feel confident that you can find on your own. It's totally up to you. Okay, do we have any more questions? No, I think that's it. Thank you all for joining today. It was great to be with you, and I hope you learned something about our analysis tiers, base, core, and premier. Please feel free to reach out to any of us here at TokuBio, visit our website, toku.bio. And if you're in the market for mass cytometry or spectral flow cytometry, We hope to hear from you soon. Thanks and have a great day.